Give it a couple minutes. Hopefully we'll get some more people. Give it just a couple more minutes and then we'll get started. Give people a chance to join, but I also don't want to make people who are already here have to wait, so. This Kahoot is gonna be it's not organized, so like the other one that we went over last time was like set up by like kind of topical, if you think about like the lab one, right? So that's set up by topic that we were covering. This is just gonna be questions from unit 3, and some of them are mixed around and some of them are kind of like together. I think it's gonna randomize them, but it might not. It's supposed to, but we'll see. Chat, so I'm ready for it. All right, so let's go ahead and get started. Jesus. What that was. Get started. Get going with this. Okay. Remember, you can always join in with the pin from kahoot.it or through the app. If you have the app, or just type in the pin number. All right, which of the following terms is used to describe removing all microbes, including all vegetative and endospores? All right, so this is correct. This is going to be sterilization. My husband was recently talking about how he wants to get into brewing, which I don't know, but whatever, that's what he wants to do. But he was talking about how they sanitize things, and I was like, But wait, like if you're gonna be, putting, stuff and keeping it for a while, like on a shelf, like, don't you want it to be sterile? And I was looking into it, and it has something to do with like the pH, and basically Clostridium was what I was worried about. I was worried about botulism. But yeah, it's the pH. As long as it's lower than 5, it should get rid of stuff like that. But my point is, the reason I'm concerned about, botulism and stuff like that is because I know that if it's a sanitizer, then it is not sterilizing, because that is a different thing, right? So, but yeah, just be aware of those terms and just be able to know them from one another, right? Okay. Which term refers to the removal of microbes from a living surface, such as skin? All right, and if we're targeting the skin specifically, and we're trying to remove the microbes in generally from the skin, it's going to be asepsis. Decontamination has to deal with removal of microbes, yeah, but it has to deal with removing them mechanically. Specifically mechanically, right? So it's a little bit more specific. So because it involves moisture and heat, boiling is an effective way to sterilize things. Is that true or false? Basically, our question here, right, is asking if boiling can sterilize things. And it does not sterilize. If you're boiling, generally, normal temperature of boiling, 30 minutes of boiling, just water, no added pressure. It will not sterilize. It won't get rid of the endospores. But it is fine for sanitizing. Okay, autoclaves sterilize items by using heat, moisture, and pressure. Is that how an autoclave works? All right, so this one is true. That is how autoclaves are going to achieve sterilization. And autoclaves can sterilize as long as you're reaching a certain temperature, and there's moisture present and the pressure is present. Remember, the moisture is important as well. It helps to denature the proteins better. All right, true or false? Non-ionizing radiation, like UV light, is able to penetrate items. Okay, so this is false. Non-ionizing radiation, like UV light or UV light specifically, UV light can be used to sterilize even some surfaces, but it's not ideal because it doesn't penetrate anything in any way really effectively the way that ionizing radiation can. So yeah, so in this case, non-ionizing radiation like UV light, it will not be penetrating things. So downfall of it. Which term means that the agent will actually kill the microbe? Yeah, so this one's pretty much self-explanatory. Is that, what is that? Yeah.
Yes.
Anyways, microbicidal, just like it says on there. Like homicidal, and we've learned about this, we talked about bactericidal and all that when we were learning about Kirby Bauer, so this isn't a new concept, so you guys pretty much got that one down, not surprising. All right, how do surfactants work to control microbial growth? All right, so they are amphipathic, like phospholipids. That's exactly right. If you guys remember from chemistry, I actually don't know if you covered this in chemistry. I'm just assuming you did. But part of what we would be taught in kind of biochemistry stuff, when we have something like a phospholipid that is amphipathic, you have the charged head, whether it's positive or negative. A lot of times in our surfactants, we're talking about a positively charged, like a quaternary ammonia compound. But the heads that are charged will kind of hang together and associate with the water that's polar outside, and then the non-polar tails on the inside. And you can even create sort of like something very similar to a lipid bilayer and to contain things. That could be containing oils if we have the tails on the inside, or it could be containing other things, even whole microbes, if we're creating those kind of lipid bilayer things around them. Those would be called micelles whenever we do that. Have you ever heard of micellar water or something like that? But in this case, we're talking about something like a quaternary ammonia compound creating the micelles. So that's how they work as a surfactant. So we see the formation of those bubbles because we're getting them all forming those little kind of bubbles, really, with their charged heads and stuff on the inside. All right, cool. All right, which of the following antibiotics work by affecting the cell wall? Now we're getting into it. All right, so penicillins, this is what the answer's gonna be. Now, I highly recommend for understanding like the antibiotics and knowing which one falls into which category, sort them out by which part of the microbe they affect. Is it gonna be the cell wall? Is it gonna be the, the nucleic acids? Is it gonna be the membrane? Is it gonna be the protein? Or is it gonna be the folic acid synthesis pathway? So those are the 5 that we have in just the bacteria. Then I would have another category for the fungal, antifungi, and then the ones for the protozoa, and then the helminths, right? Missing something, I don't know, might be. But anyways, antivirals maybe, but I mean, we don't really get into the antivirals too much. I would definitely be aware of Tamiflu and Relenza and how they work against flu. That's an important one, especially because it's just going to come back around when we learn about the flu anyways. So how those work to help inhibit, the neuraminidase aspect of the flu. We can get into it, we can talk about it now if you really want to, but remember neuraminidase, we have H1N1, for example. H is hemagglutinin, N is neuraminidase. They're 2 major aspects of the structure of the virus and how they're characterized. So the neuraminidase helps for entry into or budding out of the host cell for the virus. So if you can block that, you could effectively block the infection as long as you're getting to it, relatively early on in the infection. So it might be too much for it to handle if it's already a full-blown infection, though. So the sooner the better when treating with those. Anyways, so I would divide it into those categories and be able to recognize that if you see penicillin, it's going to be cell wall, or if you see cell wall, which will go to the cell wall antibiotics, be able to list off the ones that we have on the study sheet. You don't need to go beyond the ones on the study sheet, though, really. All right. Okay. All right, which of the following antibiotics work by affecting the cell membrane? There's really only one that you need to know for this category. All right, so this one is polymyxins. This is kind of, you can say polymyxin is like more a category of it, but you don't really need to even worry about that. If you see cell membrane, the ones that are going to be affecting the cell membrane structure are going to be polymyxins. You guys have heard of Neosporin. I know I talked about it in the lecture, but yeah, Neosporin. I mean, that's kind of important to understand why that is effective and how Neosporin works in order to prevent, your wounds from getting infected and help them heal, because we're not letting bacteria affect the healing process. There's 3 antibiotics, a triple antibiotic cream. You can just buy generic and they'll just be called triple antibiotic cream, right? It's got neomycin. That mycin is a hint. Generally, most of the mycins, not all of them, of course. But a lot of the mycins are going to fall into that protein inhibit category. So affecting how the ribosomes are making proteins, right? So we've got neomycin affecting protein synthesis. We've got the polymyxin that is going to be affecting the membrane structure. And we've also got bacitracin, which is going to be affecting the cell wall. So all three of those things targeting all at the same time makes it very effective for preventing, spread of infection, targeting all those things instead of just one. You go take one of these antibiotics orally, it's usually just one that you're taking. So you can see how it could be more effective with three. Alrighty, which of the following antibiotics works by affecting the folic acid synthesis? Oh gosh, the dog's eating. All right, so folic acid synthesis. The giveaway here is going to be sulfa. So if you see sulf in it, then that's going to be a giveaway. The other one that falls into this category that doesn't directly say sulf usually comes along with sulfamethoxazole, and that's trimethoprim. So if you see trimethoprim anywhere, just remember trimethoprim and the sulfas for the folic acid synthesis pathways. If this is a great target for our antibiotics because humans do not synthesize folic acid. We have to get it from our diet. And I'm sure that you guys know of folic acid in its other kind of form as it comes biologically as folate. A lot of people will learn about it as methylfolate. That's still folate. It's just kind of with a methyl group to make it more bioavailable. If you guys have ever heard of the MTHFR, what that would look like, MTHFR, that is the receptor for methyl folate, basically. We learn about it as L-methylfolate, right? And that's just the version of the methylfolate that we can use biologically. When you have certain mutations in that receptor, the MTHFR receptor, in those genes, it can affect how your body is going to be able to, make new cells and things like that. Folate is just essential. It's essential for building nucleic acids and for making amino acids. Any of the molecules, the large macromolecules in your cell can depend on folate. Anyways, you have to be able to absorb it effectively and use it effectively in order for you to make new cells. We can't make it in our bodies. We have to get it from our diet or from supplements. Like if you're pregnant, they recommend that you take folate. They also say that low folate levels are associated with depression and low energy and things like that. I'm not saying that everybody needs to go start taking a folate supplement or anything. Go talk to your doctor first, but anyways, so we have to get it from our diet somehow. And that, like I was getting to the MTHFR gene, it's associated with cancer, with a lot of cancers actually, and specifically with breast cancer most famously. So, but yeah, so those are linked for us, just a little tidbit of information. But for bacteria, they make their own folic acid. They can use things to generate folic acid. So they have pathways that they go through to make folic acid. And we can stop that process in a lot of different places by inhibiting the enzymes involved in those processes. We would say, as we learned in Unit 2, would be competitive inhibition. So we're binding at the actual active site typically with these drugs. They bind at the active site so that, whatever middle precursor can't. And so that's how we will block folic acid synthesis in bacteria. So all the drugs that we have that are effective at doing that are sulfa, something with sulfa in it. Okay, any sulfa, if you see sulfa in the drug name, that is what falls here. Okay, and then trimethoprim also falls into this category. Okay. All right, which of the following antibiotics work by affecting protein synthesis? All right, so it certainly doesn't cover all of what we would be seeing with our, protein synthesis inhibitors. One of the things we learn about with protein synthesis inhibitors, there's a big group called the aminoglycosides, and that will include things like streptomycin or gentamicin and those sorts of things. And then we have these groups that are named with the cyclins. So that's going to be tetracycline and glycylcycline typically. So that's how we will be dividing out our protein synthesis. Aminoglycosides, including the mycins, not all of them, right? Not all of the mycins, but a lot of them. And then tetracycline, glycylcycline. All of these guys are going to be affecting the function of the ribosome and how it works to make proteins during translation. So that's why we had to learn about that stuff before we got into this. All right, which of the following antibiotics work by affecting nucleic acid synthesis? This is a group. I'll let you know that. Okay, so the nucleic acid synthesis group is going to be the fluoroquinolones. The fluoroquinolones are the group of drugs that are going to affect nucleic acid synthesis. The drugs that literally fall into this category are going to be the floxacins. So ciprofloxacin falls into this category. Floxacin is a fluoroquinolone. Anything floxacin is a fluoroquinolone. There's other ones that will fall into that category, that and infloxacin as well. But cipro tends to be the one that most people are familiar with, so that's why I use it as an example. But be aware, if it ends in floxacin, it will be a fluoroquinolone, and if it is a fluoroquinolone, it will affect nucleic acid synthesis. Remember the aminoglycosides, I just mentioned those are going to affect the protein synthesis, and then cephalosporin and carbapenem both fall in the cell wall category. Carbapenem is a like last line of defense medication. Which of the following medications are used to treat the fungal infections? All right, a large number of the azoles will fall into this category. The ones that you see medically a lot are going to be ketoconazole, miconazole. Those two are the biggies. So miconazole is the one that you would buy over the counter to treat a lot of yeast infections. A lot of the 7-day treatments, for example, will have miconazole in them. And then ketoconazole is a pretty common one as well. Fluconazole is one that they often give orally for things like yeast infections or skin-based fungal infections as well. Miconazole is also what they might use for like athlete's foot fungal infections. The other fungal medication that I would be aware of other than azole-based medications, that would be amphotericin B. Amphotericin B is pretty widely used as like a second-line treatment if the other things aren't working. It's a little bit more hardcore, but it is another one I would be aware of. So let's move on. Which of the following medications are used to treat malaria? So based on our knowledge of drugs that we might use to treat malaria, let's see if we can pick out the right one. All right, so yes, it's going to be chloroquine. So this is based on quinine, which if you guys were, paying any attention to the lectures, quinine was the old version of what they would use to treat malaria. It caused blindness and stuff, but it was pretty effective at preventing malaria whenever people were traveling in areas that were endemic for malaria. And then they started mixing it with gin. As a gin and tonic. Tonic water contains quinine in it. That was the way that they would treat malaria or prevent malaria as prophylactic, right? So fevers are dangerous and do not provide benefit to the patient. Is that true or false? All right, so this is false. Obviously, fevers, raising the temperature, getting our metabolism working, activating our cells in response to an infection. That high temperature can also inhibit the growth of a lot of pathogens and help inhibit their enzymes, prevent them from being able to work. So it helps our cells, but inhibits theirs. It also, involves signaling pathways that will help activate the immune system and help hasten the response of the immune response. So it does have its place, of course, as I say in the lecture. There is such a thing as too high of a fever. You should discuss that with your doctor on how you feel about that for yourself and for your children, and when you think that should be treated. All right, true or false? Endotoxin is a pyrogen. That involves you understanding two things. What is endotoxin and what is a pyrogen? All right, so endotoxin is one of the more potent pyrogens that we are aware of that's coming from the actual microbe itself. There are two different kinds of pyrogens, stuff that comes from the microbes or the bad guys, the pathogens, the infection, and then the stuff that is coming from our signaling from our cells. So pyrogens are anything that's going to be inducing the fever response. So endotoxin is very, very potent at this. Now, what is endotoxin? If you don't remember, endotoxin is basically lipopolysaccharide, or a molecule that is found on the outside of the outer membrane of gram-negative bacteria. All gram-negatives have it, and it doesn't belong in like your bloodstream, for example. It is a PAMP. It is a very effective PAMP. Remember, we're going to talk about PAMPs here in just a little bit too, so I'm not going to give that one away. But it is a PAMP, and it is an effective one, and it will signal your innate immune system to start that pyrogen, that fever process, the inflammatory process. All right, the morbidity rate of an outbreak refers to how many deaths have occurred in the area in a given time frame. Let's hope that I changed this one. I think I had the wrong answer for this one, so hopefully I updated it. All right, so the morbidity rates, this one is false. I'll tell you why. Morbidity rate is not mortality rate. Mortality rate is what is talking about the deaths in a given time frame, right? Morbidity is the same thing as, looking at how many cases have appeared within a certain time frame. So if we're talking about morbidity and mortality reports, which is why we're even talking about morbidity, and there are morbidity and mortality reports, you can just automatically in your mind should be able to say morbidity must be different than mortality then if those are reports that we're having. Morbidity is essentially incidence, right? So it's talking about occurrence of that illness within a certain time frame. How many outbreaks we've had in the last month, for example. Which I'm sure will come up. Incidence, there we go, refers to the number of new cases of an illness in a specified time frame. Is that true or false? Incidence. Okay, so this one is true. Good job. Like I just said, incidence, that's going to be a specified time frame, right? How many cases we had pop up in the last month. If we're talking about the whole total outbreak, like let's say for COVID, since this outbreak of COVID began, how many cases have we had up to now? That's prevalence, okay? Just so you know the difference. Think of it as, I remember it as incidence. It's talking about an incident, like, and that would be a specific time frame. That's how I remember it. If that helps at all, probably doesn't. All right, true or false? Phagocytosis refers to a cell engulfing material or other cells outside of it, bringing it inside to be broken down. Is that what phagocytosis is? All right, this one's true. Phago being eat, cyto being the cell. So the cell is eating things up. And that is exactly what this is describing, the cell engulfing material or other cells. and bring it inside to be broken down. Whenever we create the phagolysosome, right? So don't forget the steps of phagocytosis. I'm not sure if I have any questions over it, but don't forget it. Which of the following is the most numerous of all the white blood cells? If we do a CBC, and we might have the overall white blood cell count, but which one is likely to be the highest in a human blood sample? This is an important one to know for medicine, as well as you'll learn this again in physiology if you haven't taken physiology yet. It's going to be the neutrophils. They make up about 55% of your white blood cells. It's quite a lot. And they're important, right? So we learn about the neutrophils and their role in the innate immune response. They go around performing phagocytosis, right? They'll go and recognize PAMPs, which is just anything that's signaling them that this is bad guy. They go in, they eat these things up and digest them. And then they essentially poop out what they digest. They just, exocytose or release all what the digested material is out. They do not present it, as we learn in the innate response and then into the acquired response. They are not antigen presenting cells, but antigen presenting cells like macrophages in the area will come and clean up the poop and the mess that the neutrophils left, and then they can present it. And they'll present it on their MHC2, right? Because they're finding it on the outside. So neutrophils are extremely important in that response because they can be there pretty much right away. We don't have to stimulate them quite as specifically as we might other cells that can respond. So it's essential that they be able to respond to infections. Macrophages are monocytes that have migrated into tissue. Is that true? Is that what macrophages are? All right, so this is true. Macrophages and dendritic cells are basically monocytes that have moved out of the bloodstream and into their respective tissues to differentiate. Anytime we've moved out of the bloodstream as general monocytes and gone somewhere to differentiate, we get a new fancy name. Macrophages and dendritic cells fall into this category. So monocytes are like what they start out as, essentially all the same thing as far as the functionality is going to go for the long term. All right. And depending on what signals they get, it's gonna tell them where to go and what kind of macrophage they'll be when they get there. So which cytokine, this is a cell signaling molecule, a cytokine, enables a virally infected cell to let its neighbors know there is an impending viral infection? All right, so you're infected with a virus and you're gonna send this signal to your neighbors. This is like my favorite story, little example that I give. All right, so this is the effective situation of a burglar coming into your house. The burglar is in your house, let's say. He's made it that far, and he's already in there going through your things. You're hiding in your closet, and you have your shotgun, or maybe you haven't gotten your shotgun yet, right? You're hiding in your closet, but you do have your cell phone. So you can call 911, and that's fine. We have to wait for the police to get there, right? And that's going to take a while. That might be your CD8 T cells. However, while you're waiting in the closet, you can call your neighbor and let them know, hey, Tommy, we've got a burglar in our house, and he's in our house right now. I'm not sure if things are going to go well for us. However, he's probably heading to your house next. Get ready. So Tommy can grab his shotgun and wait for the burglar, even though he knows maybe it's not safe to leave his house. That could be worse. So he'll wait there for the burglar there and will be prepared whenever the burglar shows up at his house. That is interferon. Okay. You can also think of it as it is interfering with that burglar's intentions. So the burglar in this case is the virus. So this is just allowing the cell to let the next cell know there's a virus. Get ready. So that's interferon, abbreviated as IFN. All right, true or false? PAMP stands for pathogen-associated mediator protein. Is that true or false? You've already heard me talk about PAMP, but what does it stand for? All right, so we kind of split on this one. PAMP, if this is false, it stands for pathogen associated molecular pattern. So when we talk about molecular patterns, we're talking about things like endotoxin. So if gram negatives are coded with LPS, lipopolysaccharide, the presence of that molecular pattern, that's what's going to be triggering the response. This is a general marker for bad guy. Does the presence of endotoxin, is that specific to any specific pathogen? Absolutely not. It's not different for the pathogens. All of the pathogens, the gram-negative pathogens, let's say it's E. coli or Salmonella or Shigella, they're all gram-negative and there's even more, but they all have endotoxin on the outside. And that's not going to differentiate between them. So this is an innate response. It's something that your innate system can just see and say, this is bad guy. So the presence of endotoxin is one, or peptidoglycan, that shouldn't be around in your bloodstream, right? So if cells that are, responding to that see just peptidoglycan around in areas that it shouldn't be, that's another PAMP that it can recognize. This shouldn't be here. Double-stranded RNA, we don't have that in our cells. That's a weird thing to see. Viruses can have it though. So that would be an automatic PAMP. Does it recognize a specific virus? No. Does it create antibodies against that endotoxin when we see the PAMP? No. It just signals the innate response. So these innate response cells can just say, bad guys here, start killing anytime that they see those things. All right, what triggers the classical complement pathway? All right, antibody is the correct answer here. It can also, there's two other pathways, right? The lectin triggered pathway where we recognize certain sugars, the mannose basically on bacteria that we wouldn't see on our cells. Like a PAMP, basically, yeah? And then, that would be one, and those are the same thing, the lectin and the bacteria associated mannose. Then we have things like the lipopolysaccharide, which is the alternative pathway, that just falls into that category. LPS or endotoxin falls into that category. But yeah, so those are the three ways we can trigger complement. What the heck is complement? Complement is little proteins that are always found in your bloodstream, constantly there. They don't have to be, triggered to be made or anything like that. They're just always present in your body. And when they are triggered in one of these pathways, then they will start to they can be involved in all three things. Inflammation, they can trigger inflammation, so the inflammatory response. Before anything else sees anything, we can start the inflammatory process going right away and, signal our innate response to kick into gear. The other thing that it can do, we have the opsonization, which that's a fancy word that's going to come up here in just a moment, but basically creates molecular handles for your phagocytes to grab onto. Remember how we learned about how it's hard for our phagocytes to eat bacteria that have capsules? Well, opsonization gets around that. Opsonization creates little handles for the phagocytes to grab onto so that we can eat cells that have things like capsules, makes it easier for phagocytosis to occur. And then we have the membrane attack complex, which is kind of the neatest thing that complement can do. It basically pokes holes. It creates little protein holes so that the contents of the pathogen will leak out and then it can't, do its process effectively. It will die eventually. So it's pretty cool. Complement. What is opsonization? Let's see if you were paying attention. All right, so this is the little handle. So we kind of already went over this. So I'm not going to go over it right now. But you guys got it. All right, next one. What is the purpose of CD4 positive T cells? Now we're getting into adaptive immunity. All right, so the CD4 positive T cells are helper cells, and basically they're going to activate B cells. B cells come up, you know, with some sort of thing that they found on their little MHC2. The T cell will come up and say, yep, that looks good to me. I match up with this, so you are good to go. I'm going to activate you. So everything has to have a checkpoint saying, yes, you can be activated, because a lot of our immune response is really damaging to your body if it is not done correctly. Look at what we learned about in chapter 17, right? So if we look at all those things that can go wrong with the immune system, we have to have it all in check. So, so they help with that, as well as helping to moderate your CD8 response. They give extra signaling to the CD8 cells. They need it because the CD8 cells are going out to kill your cells that are infected, right? They better be infected. So part of that Th1 type of CD4 cell, the Th1, think of MHC1, that's going to be stuff from inside, that's going to be the CD8 response. And so in order to get those CD8s activated appropriately, they have to have cytokines, signals coming in saying, yeah, go ahead and kill your own cells. Basically, got to keep that in check, right? Everybody's saying, yes, you have the go ahead. We're all seeing the same bad guy. We all agree that this is the bad guy. All righty, this could be more than one choice. So what happens when B cells are activated properly? Try to read your choices. I'll tell you that it's two of these. All righty, yes, you guys pretty much getting this. This is in fact going to be proliferate and differentiate. So there's that. But what do we differentiate into? It's actually going to be three things mainly. So our plasma cells, that's going to be our antibody producing cells, and our memory cells and our regulator cells, which we don't have listed here. But that's pretty much what you're going to get anytime you activate any of these adaptive immune cells, right? At the bare minimum. All right. This could be another one with more than one answer. What function or functions does antibody serve? Select all that apply. So be careful and read them all. Okay, so you guys did great on this one. It's gonna be everything but the teal one. Antitoxin, the antibody interacts with the toxin itself. This is exactly how your vaccines to tetanus and diphtheria and pertussis work. They attack the toxin, not the bacteria. So those antibodies that you're generating are against the toxin, which is the dangerous part. So for those diseases anyways. Then we have complement activation. We already talked about that, but antibody is the classical pathway for activating complement. If you're making antibody and that is present, that's going to activate complement, which we already talked about what that does for us. Opsonization, so that is our little molecular handles to help with phagocytosis. Agglutination, that's causing the things to stick together. So the antibody is reaching across one way and another way and grabbing across, and then we've got all these antibodies helping stick all these cells or viruses together so they can't go do their thing. Sticking together like a clot of red cells. And then we have neutralization. That's going to be a specific antibody interaction with like the spike of a virus so that it can't interact with its receptor on your surface. So it interacts with the key so that it can't get into the lock. So it's a very specific interaction. There's another one that isn't listed on here that we need to know about, and that is coding. And it's very similar to neutralization, but it's more general. So we're just binding to the molecule on the surface of the cell or the virus and just blocking it, just completely coating it all over so it can't do whatever it's trying to do. So it's more general blocking than the specific one that we see with neutralization. Okay. True or false? Upon reactivation, memory B cells will produce primarily IgG. Is that true or false? All right, so this one is going to be true. IgG is our workhorse antibody, and we got to think about, IgG basically in our memory B cells, so those plasma cells that we have. When they get reactivated again, whether you had seen it from a vaccine or you'd actually gotten ill from whatever the thing is, once these memory cells see it, they just immediately start cranking out antibody. And the antibody that they crank out is IgG. IgG is super effective, the most effective antibody that we have. And we can make it in such high quantities right away that your body reacts with the antibody response so quickly, you never even knew that you saw the thing. At least that's the hope with a lot of these, right? So with our vaccines anyways. So that's why, you could be around somebody that had measles or chicken pox or something like that and never even know. You don't get itchy, you don't, nothing. Super fast. IgG is our workhorse that does that for us. All right, true or false? Cytotoxic T cells are CD8 positive. Okay, so this one is true. Good job. Cytotoxic, these are cytocell toxic to cells, T cells. So that's their whole function, is they're going to go around killing your infected cells. So self cells that have, virus or something like that, right? Taking them out so that they can't spread it to somebody else. It's asking us, are they CD8 positive? What does that really mean? It means they have a marker on it that we call CD8. And they do have that marker. So we've identified all of these with antibodies, believe it or not, fluorescent antibodies. And they're just named. They don't have a purpose to their name, but just you do definitely need to know CD8 and CD4 and the difference, right? All right, which immune cells produce granzymes and perforins? All right, so this is going to be the CD8 T cells, our cytotoxic T cells. This is what makes them cytotoxic. They secrete perforins that poke holes or perforate the target cells, and then granzymes that go in to break down the contents of the cell, and that eventually will lead to the cell undergoing apoptosis or cell death. All righty. Regulatory cells are present during the immune response to make sure the response is as strong as possible. Is that true or false? Is that what regulatory cells do in the immune response? All right, so this one's false. Yeah, so they're going there to make sure that you don't overreact, to keep things in control. When that stuff goes out of control, when we don't have a proper regulation of these responses, that's whenever you can see things like toxic shock syndrome. So having an overreaction to something. So super antigen would be what does that, right? Okay, vaccines activate the immune response by a completely different means than the pathogen would. Is that how vaccines work? All right, so this one is false. Vaccines act the same way. The whole point is to activate the body in a way similar to the pathogen, so that when you see the pathogen, you will react the way that you should, right? So it will be effective. They use the same system, same immune system. So which of the following is an example of a type 1 hypersensitivity? All right, so this one is allergies. Type 1 allergies. So allergy to ragweed. So we think of this as a typical, atopic reaction. So localized. Remember, anaphylaxis is going to be the systemic version of that. I'm going to ask you about what antibody. later. Which of the following is an example of a type 2 hypersensitivity? Couldn't spell apparently. Okay, so this one is hemolytic disease of the newborn. I know we didn't really get into talking about toxic shock syndrome. We didn't get into talking about Arthus reaction. We did talk about anaphylactic shock, but we should know that the hemolytic disease of the newborn, that's the one when we talk about ROGAM. That's the one that you need to be aware of for the type 2. Of course, rejecting your, a blood type that doesn't, jive with your own, that would fall in that as well. But the hemolytic disease of the newborn, pretty important one. To be aware of going into nursing, right? Whole point of ROGAM. So which of the following is an example of type 3 hypersensitivity? Okay, so it's a mouthful. Acute post-streptococcal glomerulonephritis. And it is kind of what it sounds like if you know what glomerulonephritis is. Talking about nephritis, that's talking about inflammation in your kidney. Glomerulo is talking about the glomerulus in the kidneys, such structure in the kidneys. Okay, so we're talking about things affecting that filtering aspect that your kidneys do for you. So post-streptococcal, you had some streptococcal infection. It could have literally just been strep throat. But the antibodies that you made against that strep infection are getting caught up in they're binding with each other and with antigen and creating these immune complexes. That's the whole definition of the type 3 response. So immune complex formation, getting caught up in the kidneys. When they get caught there, your immune system still sees them as an attack on a bad guy. So it starts the inflammatory process and attacking everything nearby and causing tissue damage in the kidneys themselves. It can be deadly. It's a very, very serious response. If you have had, this acute post-streptococcal glomerulonephritis, you would know it pretty quickly. So it's not anything to mess around with. Alrighty. Well, this could be more than one. Which of the following is an example of type 4 hypersensitivity? Type 4. I don't remember why I put a wolf on this. I like wolves, so that might have just been the only reason. So, yes, so type 4 is dealing with graft rejection and poison ivy. So there's other ones that are going to fall into that category as well, but of our choices here, graft rejection, basically your body interacting. So you get a new kidney, your body sees that kidney's MHC1 that's just chilling. It's just MHC1. All of your cells have MHC1. Everything has to have MHC1 because that's the signal of the viral infection. Most tissue can become infected with a viral infection. This is how you would signal that on. So MHC1 is just chilling, but it's from somebody else. And the way that it looks to your body, it attacks it. And that's going to be with your CD8 response because it's MHC1. So this is type 4. Anything dealing with your CD8 response, your T cell response, cell-mediated immunity. So that's going to be a delayed type response. That's type 4 hypersensitivity. All three of those things. Cell-mediated. That's because the cells that do the work, the T cells, the helper cells, as well as the CD8 positive cytotoxic cells, they're the ones that actually do the work. So it's cell-mediated. It's delayed because you have memory cells for your T cells, but you don't have them in spades like you did during an infection. You don't. You only keep a certain amount of memory cells. That means you have to repopulate. They're already activated. They're mature versions. They don't have to go through selection process again, but it still takes time for us to build back up that immunity when we see that bad guy again. So it takes time. So that's why it is delayed type hypersensitivity. Okay. So that's why we have those two things going. Poison ivy, I already told you guys that I was pulling, you know, the weeds, and I was afraid that it was poison ivy I pulled, and I wasn't going to know for a couple days, because that's how this works. It's delayed type hypersensitivity. Same reason why when you get your tuberculosis skin test, you go back in a couple days, not like, you know, 5 minutes later. If you were allergic, you would have a response. If it was true allergy, type 1 sensitivity, then you'd have that response within minutes. If it was your antibodies, your IgG that was responding, that we were looking for, then that would be within, you know, minutes. You'd have that reaction. It's not, though. It's your CD8 response that we're looking for. And so we look for that a couple of days later to see if we have an inflammatory response from that. And if you do have an inflammatory response, it means you have seen tuberculosis at some point in your body.
So does anaphylaxis fall into a specific type, or can that happen with multiple types?
Generally speaking, you're really only going to see anaphylaxis with the type 1
Okay.
because it's going to be the IgE, and it's going to be that response associated with IgE. And it will have to be antibody every time. Whenever we're talking about the type 2, it really is going to be anything but IgE, more or less. So IgG or IgA or IgD, any sort of reaction with any of the other IgGs, or excuse me, immune globulins against a blood type. So, but yeah, it won't be IgE related. Won't be that. Make sense? Hopefully that, you know, parses that out. All right. Let's go on to the next question. True or false? HIV is an example of a primary hyposensitivity. Is that true or false? We're talking about HIV infection. I'll be clear here. I'm not trying to trick you with the HIV versus AIDS. So you think of HIV into AIDS. Is that a primary hyposensitivity? HIV developing into AIDS. That is basically going to be a secondary hyposensitivity. Looks like most of you guys got that one, right? So primary, what does that deal with? Genetically related hyposensitivity. So having immune dysfunction or low immune response genetically from genetic mutations. Born with it. All right, you run an ELISA to check if your patient has antibodies in their serum against RSV, respiratory syncytial virus. What kind of test is this? What kind? Does your patient have the antibodies? Look at that. All right, so that's indirect. So definitely be aware of your indirect versus your direct. Specific versus sensitive. Those sorts of things. We've already seen these terms before, so they're not news. And then the different test types that we talk about, because I don't really go over them that much in here. Just be aware of how you would be applying them and why. And definitely be aware of, if I say, oh, you're going to be performing an ELISA test on somebody, you should definitely know that that falls into the immune category, right? So not the DNA or genetic category, or even the physiological category. Because remember, that was our other category, the physiological tests. Those are just like the crap that we're doing in lab right now. So the citrate test and stuff like that. All righty. What is the term for the ratio of the concentration of drug that is toxic to its effective dose? So the amount of drug that would make it toxic given to a person versus the amount that you would need to take for it to be effective against the bad guy. There's a term for it. So we're talking about just the ratio of these things. We're talking about the therapeutic index here. So to run back through the concepts of what these other things are, because they are relevant within the exam, selective toxicity is referring to just the concept that when you're giving somebody a drug, you want to be as, you know, toxic as possible to the bad guy and least toxic as possible to the person that's infected, right? The host. That's just the concept of that selective toxicity. Therapeutic index. If you take, you know, 50 milligrams of tetracycline, and you can take up to that, and then you start having ill effects, so toxic effects, right? So take up to 50 milligrams, but you have to take 45 in order for it to be a minimum of 45 in order for it to be effective against E. coli or something. That's really, really close, you know, that might be dangerously close. So you might want to go with another antibiotics. So the closer they are together to similar, so 50 to 45 is very close. The ratio is very close to 1. You want to steer away from the 1. You want them to be as far apart as possible. Very toxic. So if you can take up to 50 milligrams of tetracycline, but you only have to take 1 for it to be effective, you're pretty much safe at that 1, right? So you probably have a little bit of leeway as far as that goes. So that's what that is referring to with that therapeutic index, all right? The ratio of the toxic amount versus the effective amount. The zone of inhibition, remember that's going to be the clearing area that is surrounding the disc on the Kirby-Bauer plate. And the minimum inhibitory concentration is the minimum amount that it will take in order to inhibit the growth of that microbe. It could be referring to inside of a patient, but usually we're talking about for in the Kirby-Bauer assay, right? So the minimum amount, so it's going to be the outside edge determined by the amount of the drug at that outside edge of the zone of inhibition. You can calculate that. We don't have to for this class, thankfully, but the lab will do that for you whenever you have that issue. So those are the differences. Alrighty. Taking an antibiotic can clear out your normal biota. That will leave the hardy ones behind to overgrow. There's a term for this. What is it? Exactly. This is super infection. We see this with C. diff. People taking a lot of antibiotics, the C. diff will survive because it's got its endospores, and they travel quite easily. So, but anyways, it'll overgrow. Remember, we talk about, well, I say it a lot anyways in the lectures, which is the stadium seats. All of your good microbes taking up stadium seats, some of them that are typically good in small numbers to take up stadium seats, like C. diff might be part of a stadium seat. However, you kill off everybody else, but only the C. diff is left. They're just going to multiply and take up all the stadium seats. And then you've got pseudomembranous colitis, and that's a problem. So that'll happen. Same thing happens with yeast infections whenever you take antibiotics for UTIs, for example. Can happen. So that's called superinfection. Usually comes from yourself, from your own biome. So true or false, all bacteria are resistant to antibiotics that are made by other bacteria. Okay, exactly. So once you guys got this is false, it's false because this is literally where the concept of antibiotics even came from, right? So some bacteria make antibiotics to kill other bacteria, and that's like the point of them. So yeah, so this was just a little bit of getting you thinking about taking a step back to the beginning. All right, where do all white blood cells originate from? Great job, guys. That's exactly correct. Bone marrow. You just got to know that one. All right. This could be more than one. Select the granulocytes. All right, so the fills. On the exam, I'm not going to ask you about any of the granulocytes besides the fills. Mast cells do fall into the granulocyte category. I'm not going to include them. I just want it to be easier on you guys, especially because when we're looking at, you know, CBCs and stuff like that, the granulocytes, the staining procedure that we'll be looking at here, the neutrophils, the eosinophils, and the basophils, these guys are the ones I'm more concerned about, right? So the fills, so think about those guys. The granulocytes, or those the agranulocytes, that will be like your lymphocytes and your monocytes and their derivatives, like macrophages. All right, what does calor refer to? An inflammation. We had 4 Latin terms, and then we had also loss of function as a 5th one. Doesn't have a Latin term. All right, so this is going to be the heat CalOR, think of it as like CalOR if you don't speak Spanish. If you speak Spanish, then this is probably really easy for you. But if you don't, then CalOR, just think of it as calorie. A calorie is measured as the energy that's released if you burn the energy. And really burning, yes, that is what we're talking to. So the amount of energy that would take to like raise water like a degree. So it's heat energy that's being released when we're doing that. That's calorie, that's calor, okay? Heat. Swelling is tumor, redness is rubor, and then dolor is pain. What is the term for molecules that are too small to trigger an immune response on their own? All right, so the term for this is hapten. Those are the molecules that are too small. They have to be introduced in conjunction with a carrier typically in order to trigger an immune response. That's what we see with tetanus. It's a hapten, the tetanus toxin, right? Tetanus toxin. And then that's what we see with pertussis, and that's what we see with diphtheria, which is why they come together a lot of times to help them elicit an immune response to all three all at once together. It works quite well that way. So yeah, and that image was supposed to depict a man who had tetanus. Remember, tetanus is going to cause, well, we haven't learned it yet actually, but you will be learning about how tetanus causes spastic paralysis. That means that it's going to trigger all of your muscles to contract. Literally all of your muscles to contract eventually, right? Over time, if you aren't clearing the toxin out of the system. We know lockjaw, but that's just kind of where it starts. So that's what will cause the seizing up and the rounding of the back, because your back muscles tend to be stronger than like your abdominal muscles. So you will have that rounding back of the back, the hands will curve forward like this, and the toes will curve forward and everything. It's the most painful thing you can experience. You can break your own bones with tetanus. Most people, if you die from tetanus, you're going to die from suffocation because you can't breathe anymore, because even your diaphragm will seize up. So don't get your shots, right? We say tetanus, oh, no, whatever, tetanus. No, not whatever, tetanus. Get your shots. So which part of the antibody structure associates with the epitope of the antigen? Takes a little bit of knowledge of what those terms mean. All right, perfect. So that's going to be FAB. So that FAB region of the antibody is the region that has the variable region. The variable region is where we get the variety that allows us to recognize specific antigens, very specific antigens, and not just the antigens, but the specific epitopes on the antigens. And the epitopes are just the part of the antigen that the antibody is going to be interacting with specifically. All right, which class of antibody is associated with allergies and parasitic infections? I did ask the question. I did mention this earlier. You guys remember I mentioned it with Chris's question. Alrighty, so we have IgE here. IgE is the allergy antibody, interacts also with, if you have parasitic infections, it tends to be the antibody associated with those. That's what that response is meant for. And that's what the hygiene hypothesis is hinting at, that if you were exposed to, worms and other parasitic infections throughout your development, then your body would have learned what to do with the IgE response appropriately. But because we don't interact with those things anymore, we think that that is part of why we have allergies. Because if you live in a third world country and you are exposed to these things like parasitic infections, they don't have allergies, like legit. So very interesting concept, the hygiene hypothesis. These other antibodies, IgD acts as a receptor on the B cell. So it's the one that it goes around interacting with in the very, very beginning of its little fun adventures on the B cell. IgG, and allows, surface interaction with that antibody. IgD does. IgG is our workhorse antibody. We already know that. It's going to come out like in mass, and it's going to be very effective. It's just our heavy hitter, right? IgA, that is going to come out in our mucosa. It comes out as like a dimer. But yeah, they call it the secretory antibody. And then there's another one that we want to know, which is IgM, which is what everything starts out as. It's a pentamer, so it comes out as 5 antibodies kind of linked all together. It's great for starting out. It's usually not as specific. We don't make it in as high a quantity. It's just not as effective at doing the things that antibody is made to do. IgG really is the best for that. So they all kind of have, their little functions there, but that's the antibodies. Which type of immunity occurs when you develop resistance after contracting the chicken pox? You got the chicken pox. I got the chicken pox when I was little. I went to one of those parties, where your mama brings you to somebody's party because somebody has chicken pox and they want to get you to have it so that you don't get it later. Little Jimmy had chicken pox. I don't know what his name is. I had no idea who it was. My mom brought me to one of those parties and I got the chicken pox. It was awful. But now I have immunity, but I'm also hiding that virus in my nerve cells, and I'm probably going to get shingles when I'm around 50 if I don't keep up with my vaccinations to keep everything quiet. So when you actually have an infection and you develop the immunity naturally from the infection, that is natural active immunity. Artificial active would be from introducing it artificially, but you still make your own antibodies. So this would be, an example would be vaccination. Artificial passive would be going to get, immune globulin infusions introduced into your body. So you're not making your own antibodies. Antibodies are being given to you, and it's artificial. You're getting it in a medical setting. It's not being given to you by a natural means. This even includes anything, adalimumab, like, that might sound like, gibberish to some people, but talking about things like Humira and things that are related to those sorts of things, they are monoclonal antibodies. And a lot of times you can go get those as infusions. And when you do, that's what, or even when you get them as shots, if you're introducing those antibodies into your body to help fight against the inflammation, which is what they do, they help fight against the receptors on your cells that react to inflammatory cytokines. So they block that way. So those antibodies are introduced passively. You didn't make them yourself. So that counts there. Natural passive. Ooh, that's a weird one, right? So this is like breastfeeding. So you aren't making your own antibodies as a baby, but somebody's antibodies are being given to you, and it's by a natural means. We're really close. Next one. All right, why is type 4 hypersensitivity delayed? Talk about this. Let's see if we remember. Excellent. So this is going to be relying on activation and propagation of T cells. Type 4 hypersensitivity is cell mediated. Like I said, that's going to be your T cells. Your T cells are the ones that do the actual work. You have two choices here, T or B cells. Do B cells actually do the work in the fighting? No, they spit out antibody. Antibody does the work. T cells are actually engaging during the fight. So they are our cell-mediated immunity. There is a name for the B cells in case you're wondering what it is. It is humoral immunity. And the humor, your lymph in your body is called your humor. That is where your antibodies are. So that's why it's called the humoral immunity. And we have the cell-mediated immunity for the T cells. It's delayed because we have those memory cells. They aren't hiding out in the same numbers as they need to be in order to respond to an infection. They still have to multiply back out, and that's what takes time. But they're still basically matured and ready to fight. They just need to multiply. So that's it. All right, so that's it for the review. So here's you guys' podium. So good job, you guys. that's it for me. You guys know how this goes. So if you have any questions, now is the time, or you can email me and I'll try to get back to you before the exam. Don't forget your computers. Yeah. We have a couple of minutes, but yes.
I noticed one of the quizzes due date was set to 11:59 a.m. today. Was that supposed?
Yeah, so that was an accident.
Okay.
Yes, of course it was. So if you took it late, and you still can take it if you haven't taken it yet, just take it late. It won't count against you, I promise. Yeah, but you can still take it. Yeah.
Okay, thank you.
Yeah, no worries. I forgot about that. Somebody else mentioned it earlier and I just completely, yeah. Man, this semester is going by too fast. Summer always feels like it's I don't know, it's too fast. You guys have any other questions? All right, so if that is it, if you guys don't have any other questions for me, I'm gonna go ahead and log out. You guys can do the same. I will get the link posted to this if you want to play through it on your own time. And I will also post this recording once it has processed. So good luck tomorrow, and I'll see you then.